Welcome to Part 7 of the Reproductive System, page 1056, in which we pause between genders to look carefully at the embryology of gender. And we're taking a moment here to observe the fetal development stages. Technically, this is still slightly between embryo and fetus at 5 to 6 weeks of gestation. We don't stop being an embryo until week 7 or 8. So here it is sexually indifferent, which does not mean that it doesn't care. It means that it's both at this point. And so in this case, we have a clear indication of the kidneys in the pelvic region. We have a clear indication that the gonads are high in the abdominal cavity and will eventually be traveling down this little sort of railroad track here, okay, of the mesonephros, as it were. The mesonephros is the female pathway, and that's the pathway we have at this stage of life. First of all, we are all, in some ways, like the perfect flower at this point. We are both male and female. And in another sense, we are actually just female at this point because we only have the mesonephric. And with the absence of a Y chromosome, or the effective gene called SRY on the Y chromosome, all of us are destined to become female. That is the standard and the default gender of our species is female. You might take a look at this moment in now 2020, in the month of April, at the verified 50 oldest people in the world in Wikipedia. It's the first time I've seen that list of 50 oldest people up to around 116 or 17 years of age, that the oldest verified people, the 50, are only female. There are no males at this point. Historically, there was always in the top 50 something like 2 or 3 males in the top 50, but at this point in time, there's no males whatsoever. There's just females. And it indicates a kind of, how would I say, homeostatic stability here in this sense of gender. So one takes a chance, as it were, to turn off the mesonephric, which is the standard system, and then go, or that is to say, not turn off the mesonephric, to turn off the, turning off the paramesonephric, and to keep on the mesonephric system. So Wolffian ducts here are representing the in this particular case, it's going to be the epididymis, and this is the mesonephric duct, which forms the ductus deferens. These are the male ducts in this case, and that this individual who has an effective SRY gene, one of the very few genes on the Y chromosome, which is sort of the smallest of all chromosomes and the least, number of enumerated genes present there. That means that this person is going to follow the male pathway, and that the mesonephric duct will, the paramesonephric duct rather, will disintegrate, and the mesonephric that I'm referring to here as Wolffian duct is going to persist, and it's going to become the male parts. There is going to be a prostate gland, not a uterus, and so on. And we'll look at the next page for some of the outcomes. Over here, we have an individual who either does not have a Y chromosome because they have 2 X chromosomes, or they might have an X and a Y chromosome, but their SRY gene is not functional for whatever reason, or the outcome of the SRY gene might be not functional, or the receptors for the SRY gene might be dysfunctional. There's a variety of reasons that things go on this way. Also, it should be noted that this is not a switch at a particular time, in this particular time, but it's rather a constitutive gene. If you look at the current research of which I'm particularly interested, it's an ongoing housekeeping constitutive gene that's on all the time, and that in its absence it does have the effect, even in the adult, of having some feminizing characteristics. And it has in the adult over here, if there is an SRY gene, it has androgenizing types of outcomes. So it's more than just a switch. It's something that becomes effective at this particular time and is ongoing throughout life. Here we have the Müllerian duct forming, which is the paramesonephric duct. It's the mesonephric that is disintegrating. And in this individual, then, will follow the female format. She will not have the descent of her ovaries. She will have a uterus, and she will not have a prostate. Here is the uterus of interest here. And we'll be getting into the female reproductive anatomy to define different parts of it, and the broad ligament which holds everything in its place. Here is the prostate in the male, and here is the descent of the testes, usually right about the time or just before birth. Okay, and then over here we see that we have the indifferent individual. Over here, sexually indifferent at that 5 or 6 week stage, and then we begin to show developmental changes in which the male glans penis is going to take on the role of urination and will acquire urethra. The female will not. The urethral opening will remain in the vaginal vestibule, and that this will become the clitoris, the glans clitoris, and will be having its prepuce, but will not have any structural change from this point, and that the urethra is going to be something that's featured inside the vestibule between the labia minora and majora. Over here, the labia majora and minora of the male are going to come together and create individual little compartments here for the descent of the testis into each compartment, and that they'll be very well separated. And we have, as you can see, the elongation of the penis with the urethra going through and emerging at the glans penis. Okay, so that's our pathway here, and I wanted to make it clear that what is some of the take-home from this is that we have homology between male and female. We have the same cells, parts, and structures that are going through slightly different plastic, as it were, changes in order to create an accommodation for an organ that fits differently into this organ for the purpose of reproduction. And then also that we have this particular gene that makes the decision about being male. It's a gene that's on the Y chromosome, and it's a decision that becomes effective at a certain point at 5 or 6 weeks of gestation, but it is an ongoing, not just a switch of the moment, but an ongoing constitutive gene, which is on all the time or off all the time, and therefore that's creating that, at least initiating that difference and maintaining that difference across time.